Okay. So we were talking about, the bonds between our uh, bases here, the hydrogen bonds. These hydrogen bonds are weak, which allows DNA to pull apart so replication can occur, or DNA to pull apart so mRNA can make a copy of it. That being said, we're going to move on to the bases themselves. So DNA contains adenine, guanine, cytosine, and thymine. So RNA contains adenine, guanine, cytosine, and uracil. So you'll notice that adenine and guanine here are double rings, okay? Whereas cytosine, thymine, and uracil are only single ringed carbon structure or single nitrogen rings. So purines are two ringed structures, okay? And pyrimidines are one ringed structures. So cytosine, thymine, and uracil are pyrimidines, adenine and guanine are purines. And this is going to affect how they bond. An adenine cannot bind with a guanine. It's got to bind with a pyrimidine. So it's got to be adenine, thymine, okay? Or adenine, uracil if it's RNA. Guanine will bind to cytosine. So A pairs with T, C pairs with G in DNA. In RNA, A pairs with U because there is no thymine, and C pairs with G. So nothing changes there. DNA, again, double-stranded, twisted ladder, double helix, okay? It forms this helix due to the angles of the strong sugar phosphate bonds. Okay, so the sugar phosphate backbone provides these angles in which the DNA becomes this twisted ladder. Now, these hydrogen bonds between the bases are weaker. So this bond right here is strong. You want the backbone of DNA to be very strong. The bonds between the bases are weak hydrogen bonds. And those weak hydrogen bonds play the role because DNA has to unwind in order to make mRNA or to make another copy of DNA for cell replication. So each strand is going to be unique. So the way that DNA works is the A's, the T's, the C's, and the G's are all codes. Think of it as like zeros and ones for computer coding. The A's, the T's, the C's, and the G's, when they're listed in specific fashion, are different ways to source our genetic information. Okay, so our DNA is stored in the sequence of nucleotides. So it's stored in our A's, T's, C's, and G's. Okay, so since these bases are not part of the sugar phosphodiester bond, the base sequence is independent of the backbone. So the bases is what we're looking at. That's where our genetic information is stored. So any base sequence is possible. It could be like A, T, C, G, 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 whatever the case may be. All is fair game depending on what the gene sequences are. So instead of nucleotides being attracted to other bases in the same strand, because you don't want your DNA to fold in on itself and be all crunchy, they will actually be attracted to the base on the complementary strand, which helps form the rungs of the ladder. So these are bases, 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 okay? And they're attracted to the base on the opposite strand to create that double helix structure. So again, blue means video. I recommend watching them. So RNA molecules are single-stranded, okay? They actually fold in on themselves, forming the hydrogen bonds between the nitrogen bases. Exactly what we don't want to have happen in DNA, okay, happens in RNA. And it also forms bond between the bases and surrounding water molecules. Because remember, most organisms are pretty watery, okay? And these bonds can cause RNA molecules to form different shapes. So different sequences of bases, different A's, C's, G's, and U's will form different shapes, like you can see here. They're all different shapes because they're different sequences. So for RNA, our base bonding is A with U, C with G. Again, U is used instead of thymine, mostly because RNA needs to be recycled and uracil is more energy efficient than thymine, okay? So our A with U, C with G. And RNA actually played such a crucial role in early life. So RNA had the ability to catalyze chemical reactions, so speed them up or break things down, had the ability to store energy, and it also had the ability to store genetic information. So RNA is like its critical function that remains is like the creation of proteins. So RNA was this building block and it was good at all these things. However, other molecules evolved to be better. So RNA that catalyzed reaction, now it's done by enzymes, which are proteins. RNA stored energy. Now it's done by the mitochondria, baby, powerhouse of the cell, right? That's the only thing we're going to learn from biology. That's what all the memes say. Hopefully not. Okay. And then storing genetic information. RNA, now DNA does a much better job. It's much more stable. This is a slide pretty much just highlighting everything, the differences between DNA and RNA. It's one of those ones that I would save for those compare and contrast questions that tend to come up, or, why does DNA have thymine instead of uracil? Okay, why is it helical? Like, what allows it not to fold in on itself, right? So, those are things that I would keep in mind for the future. All right, so DNA is useful, it's stable, okay? It's inside the nucleus, it's protected. RNA is kind of a workhorse. It takes DNA's message and transfers it outside of the cell or outside the nucleus into the rest of the cell. DNA, princess in the white ivory tower. RNA is the one going to DNA being like, hey, look, I need your genetic code. Let me copy it. Let me bring it to the outside of the cell so we can start forming proteins, which is what we need to survive. And of course, I'm going to skip over the questions because those will be answered during the Zoom meeting. But again, remember the slides with answers is posted in your Unit 2 agenda, so you have access to it there. All right, I'm going to stop sharing here, and we're going to pick up with amino acids and proteins.